The overall goal of this procedure is to visualize enteric nervous system neurotransmitter expression in the murine embryonic gastrointestinal tract. This is accomplished by first harvesting embryos at time points of interest. The second step is to dissect the GI tract out from each embryo, taking care to separate it from the surrounding organs without damaging it. Next, the GI tract can be stored in 30% sucrose solution until multiple samples are ready for processing. The final step is immunostaining with primary antibodies to the neurotransmitters of interest and fluorophore-conjugated secondary antibodies for visualization. Ultimately, confocal microscopy is used to visualize the immunostained gastrointestinal tract.
The main advantage of this technique over existing methods is the ability to visualize the neurotransmitter identity of developing enteric nervous system neurons.
This method can help answer key questions in the enteric nervous system development field, such as when do neurons acquire their specific neurotransmitter phenotype?
Though this method can be applied to provide insight into enteric nervous system development, it can also be used to study the postnatal gut.
After euthanizing a timed pregnant mouse, transfer the uterus to a 60mm petri dish containing ice-cold PBS. While viewing through a dissection microscope, use sharp scissors to cut the uterine wall open and expose